Hi, I'm Lucie Machikova. I'm from the Czech Republic, Institute of Physiology, Czech Academy of Sciences, under the supervision of Dr. Viktoria Vlachová, and we are studying ion channels. In our study, we wanted to contribute to understanding the role of phosphatidylnositol bisphosphate, PIP2, in TRPA1 regulation. For this study, we have a contribution with biochemie membrane in nano-object from Bordeaux, University of Bordeaux, with Dr. Isabel Alves collaboration, and we aim to prove that though this part of the channel from the intracellular C terminus interacts with PIP2. As upon activation of TRPA1 channel, the calcium permeates into the channel, into the intracellular part of the cell. That in turn triggers the hydrolyzation of PIP2 from the inner membrane leaflet. We ordered two peptides of the sequence corresponding to a calmodulin binding site and to a poorly resolved part of the channel, and we wanted to prove that it binds to a model membrane of a different composition. Using the plasmon wave rate resonance, we proved that really both the peptides binds to the membrane only when PIP2 is present. Nevertheless, we weren't able to get the binding affinity. With a recently developed technique, the microfluidic diffusional sizing with the Fluidity 1 instrument, we succeeded to get the binding affinities of both the peptides. We proved that it's binding to the membrane only when PIP2 is present, and it has the lower affinity when calcium ions are present. It suggested that there might be some competition of PIP2 and calmodulin. We studied that with electrophysiology technique, measuring the currents from heterologously transfected F11 cells to mimic the native system. We proved that it's really competing with calmodulin. Next, we tried to show if that interaction with PIP2 is superficial or not. So, using the ligand docking experiment, we showed that there might be some interaction between this flexible linker that is poorly resolved and which corresponds to the longer peptide with that PIP2. And then we performed the circular dichroism experiments, and we showed that really it doesn't change much upon different lipidic environment, so it seems that there is just a superficial binding of PIP2. To show the impact of PIP2 and regulation of TRPA1, we took a look closer to this phenylalanine, which is really conserved throughout 65 vertebrate species. When we exchange it by a glycine that was supposed to disrupt this helix, and we measured the currents from hex cells that were heterologously transfected, we saw that these channels were rectified, so we suppose that PIP2 regulates TRPA1 channel at negative potentials.